Hi there, Professor Howard here again to go through the second half of reproductive histology, and that is going to be the histology of the female reproductive system. And friendly reminder, when I say female, I mean genetic female and someone who possesses ovaries and a uterus. So I'm not using gender here, I'm referring to gametic and biological sex only. So we're approaching this from an anatomical perspective rather than a gender spectrum one, because those two things are separate. Okay, away we go. So let's first look at the ovary. I'm going to pick this top one. The ovary is the site of oogenesis, and it has a lot going on, as you will see. So I'm going to first get rid of my sidebar here, just temporarily, to show you a couple things. So this is an entire ovary, and one of the features of the ovary is the medulla versus the cortex. So I'm going to outline roughly where the ovarian medulla is. And then the cortex. I want to stress when I use a bracket like this, I mean the entire layer. So I could also bracket this here or here or here. So bracket is me expressing a layer to you rather than bracketing a particular thing. So this whole layer of the ovary is a cortex. So the medulla is primarily the site of blood supply for the ovary. So you can see all these little blood vessels in here that are going to nourish the developing follicles and whatnot and provide delivery systems for hormones out of the ovary. The cortex is really the primary site of follicular development. So a follicle, and let's define this. An oocyte, which is the ovum, plus granulosa cells and thecal cells. And these two cell types have endocrine function. They produce estrogens like estradiol and other estrogens like progesterone, etc. So we'll talk a little bit about that in this video. We'll talk more about it in the lecture video. So let's zoom in. And we're going to go find some primordial follicles first. Those are generally found near the edge of the ovary. Whoa, not quite that far in, thank you. Okay, so here we have an assortment of ovarian follicles. Primordial follicles, and I'm going to write this over here. Primordial follicles have one primary oocyte and one layer of surrounding cells. We don't quite call these granulosa cells yet because they haven't popped up to become them, but they're on their way. So primordial follicles sit in the edge of the ovary, usually clumped all together in a structure that is adorably termed the egg nest. So that's this. So I'm going to circle a bunch of primordial follicles, and then I'm going to pick one or two to zoom in on just so that you can specifically see what's up. So that's the idea here. So all of these little guys are primordial follicles. Now, I want you to keep in mind as I talk, it's very important that you remember that a follicle is a multicellular structure. So if I ask you on the lab practical to identify the structure, that means what I'm asking you to do is tell me about the follicle. If I point to an oocyte itself and say, what cell is that? That's when you would say oocyte. It's a very important difference. Although the other cells in a primordial follicle are indistinct, they are still there, and you need to acknowledge that if I ask you. So I'm being very, very clear about that right up front. So this whole structure here, group of primordial follicles, is egg nest. So these are follicles which are just waiting to go. here's the deal with ovarian development. Before an XX fetus is born, during differentiation of the gonads, first spring up early germ cells that are going to become either sperm or eggs, depending on the developmental trajectory of the individual. Once that trajectory is established, in XX individuals, folks with ovaries, all of the oogonia, so that's oogonia, so the ovarian equivalent of spermatogonia, all of the oogonia begin to undergo the first stages of meiosis, and then they pause. So unlike in spermatogenesis, where there's this constant supply of new spermatocytes because the spermatogonia are always around, in folks with ovaries, that is not the case. So there's no oogonia left in the ovary by the time an individual is a neonate. They are all primary oocytes that are arrested in the early stages of meiosis, so they're not capable of giving rise to additional oogonia anymore. So that means that the population of primordial follicles that one is born with, that's all the potential eggs one will ever have, which is why the fertility window for females is different than for males. Lack of stem cells is the key there. So let's zoom in on one of these. Let me take a picture first. And then I'm going to pick one to focus on. I'm sorry, I'm kind of just scanning around to see which one I like. I suppose it doesn't really matter. Okay, I think I found one that I'm fond of. So I'm outlining, it's the smallest dot I have. I'm going to outline a primordial follicle. Notice that is the nucleus, not of the oocyte, but of a pre-granulosa cell. So when this primordial follicle begins to make its way towards development, it's going to perk up and become cuboidal, and that's going to be the first set of granulosa cells. This, I might as well make it a solid outline. This is the oocyte. So this is me just showing you, although it may not appear that way, the primordial follicles do consist of more than one cell. So I can also do the same drawing over here. So there's that. All right, so let's show you what happens when these guys perk up and begin to head towards ovulation. I'm going to draw one more of these. So these are primordial follicles. Okay. I'm trying to take a lot of pictures of what I'm drawing along the way so that you have abundant resources to look at. I know that you have your histology atlases as well, and I really encourage you to rely on those heavily, but I'm also trying to provide you with supplements. So let's go find a primary follicle. I remember seeing one nearby up and to the left that I liked. Aha, there we go. Okay. And I'm going to leave it a little bit zoomed out just for a moment because I want you to be able to see the difference. So here we have a primary follicle. And you can see this center part is the oocyte. The outer part are the granulosa cells. And this whole thing sorry, I'm just making a little space for myself to write, is an early / unilaminar primary follicle. So unilaminar, uni means one, lamina means layer, so it just means one layer of granulosa cells. So that's all that means. So another note is that I notice that I cannot see the nucleus of this oocyte. Doesn't mean it's not there, it's just that when you cut a sphere into sections, you don't always catch the nucleus of that sphere. A student during Zoom was asking about how the primordial follicles know when they're supposed to differentiate, and also why they all don't do it at once. So I'll answer the first question first. How they know is that they express receptors for follicle stimulating hormone. So let's write that down here. Oops. I wanted to get rid of that. So FSH acts on these. And also on this to encourage it to continue developing. And then the reason they don't all begin or initiate development at once is actually not well characterized by science. So that's a thing that no one knows for absolutely sure. There are hypotheses about it, but it remains one of the mysteries of ovarian fertility. And so scientists are still trying to figure it out. So just a reminder, FSH stands for follicle stimulating hormone. So if I'm talking about follicles, this is the hormone that stimulates them. Easy peasy. Okay, so I'm going to go find another primary follicle. I'm just zooming out to look for ones that I like. I like that one pretty well. I'm showing you more than one kind of primary follicle because I want you to feel confident in identifying them, whether they are an early one or a late one, because there is no specific like start and stop point for each developmental phase. They develop in a curve, so gaining size, or not linearly, gaining size in a curve, gaining cell number in a curve. So it's like the development is less like a stair step, so it's not like this, where we get primordial follicle down here, and then primary, and then secondary. But rather, you can have in-betweeny looking phases that look more like this, where primordial is down here, early primary is here, late primary is here, secondary, and then tertiary, on and so forth. So I'm showing you in between phases as well for that reason. So this is an oocyte, and now you can see there's more than one layer of granulosa cells. So zona granulosa is what we call this developing outer layer of granulosa cells, and now you can see that there are two layers. Not just one. So this whole structure we call a, let me do my white thing again so I can have a place to write. So it's still a primary follicle because you cannot see an antrum yet. I'll explain what that means in a little bit. But there are more than one layers of granulosa cells, so we call it a late or multilaminar primary follicle. Okay, so let's trash that and let's go find ourselves a secondary. Oh, that's too large. That's an okay one. what I'm trying to do I like that one. Okay, so here we have a secondary follicle. So now we have an antrum, which I'm going to label. And an antrum is a space filled with fluid. So the fluid is full of calcium ions and other cations primarily, and it's designed to be hyperosmotic to the surrounding cells so that it attracts water into the fluid space. And this provides also a space to dump hormones into. This layer around the oocyte, so here we have the oocyte, and this layer of pink non-cellular stuff between the oocyte and those granulosa cells is called the zona pellucida. Which is a glycoprotein layer between the oocyte and these cells. So they're still granulosa cells, but because they're arranged around the oocyte itself instead of around the edge, we call this the corona radiata. And then they're on a little neck, and that neck connects them to the rest of the granulosa cells. That layer we call, again, the zona granulosa. So this is a secondary follicle because the antrum is formed, but the antrum isn't yet very large. You can also see the thecal cells. So these cells right here the theca interna. And then further out we have theca externa. So the zona granulosa cells primarily produce estradiol, and they do that by taking precursors like progesterone and other estrones from the cells of the theca interna and turning that stuff into estradiol. So we'll talk more about that in lecture than we will in lab, but that's the point of those cells. So this whole thing, which I'm going to put in a big bracket. Well, hold on, I have to erase that part because it includes the thecal cells as well. So this whole thing is the secondary follicle. So let's take a little snapshot of that before we move on to tertiary. And I can see a unilaminar primary follicle in the field of view, but I'm not going to point at it because I want you to find it yourselves. But it's there. I see it very clearly. Okay, so let's go find ourselves a big guy tertiary follicle. Fortunately, there's one right here. So one thing to notice is that see how the edge of the ovary is relatively flat here, but when it gets to about this line, it starts to poke outward. That's called a stigma, and that's the area of the edge of the ovary where the antrum is going to fuse with the edge and release the oocyte. Now, here's the rub. Follicles are spherical balls of cells. They're hollow, they're full of fluid. That's what the antrum is full of. And the oocyte and the corona radiata are only stuck to one part of the wall of that ball of cells, meaning that you can have a very large tertiary follicle but you don't see an oocyte or a corona radiata or any of that because the slice simply didn't catch it. So if, let's draw in the appropriate colors here. So I'm going to take pink. Let me make myself a zona pellucida. High artistry here, as you can see. And these little dots I'm representing as being granulosa cells, so bear with me. So if the oocyte was present in this view or in this slice, it would look something like that. It just doesn't happen to be visible in this particular slice. So it doesn't make this thing not a tertiary follicle. It just makes it a tertiary follicle that we weren't lucky enough to get the oocyte in the slice of. So it still has all the same parts. So it's still got antrum, corona radiata, the zona pellucida, and the zona granulosa in the large guys is actually pretty narrow. So the zona granulosa does not grow in thickness with the diameter of the follicle. So the relative thickness of the corona radiata compared with the total size of the follicle in tertiary follicles is pretty thin. That's another way you can tell. So, oops. Zona granulosa. There we go. So I would call this big guy that I've just circled a late tertiary follicle. It's probably going to be ovulated soon. This guy over here, I would call an early tertiary follicle. It's just past the stage of being a secondary. So it is an early tertiary. It's just not quite as big as its neighbor. Friendly reminder, tertiary and graphian, G-R-A-A-F-I-A-N, are interchangeable terms. You can use either, I don't care which. Okay, and that's all I've got for ovary. So let's go. Oh, look, here's another really good secondary one. Just real quick before we move on, because it's nice to have examples of things. Here's another nice secondary. So you can see the antrum is just formed. Look at these nice thecal cells around it, but it's not super big yet. Okay. So I'm going to go back to female repro. Really quickly, I'm going to show you what it looks like at the moment of ovulation, and then we're going to move on to the oviduct. Okay. So I like a couple things about this slide. One, it's literally showing what ovulation looks like. The other is that it has several corpora lutea. So corpus luteum is the scar on the ovary that's left over after ovulation occurs. So let's look at ovulation first. So this guy right here is a big, huge tertiary follicle. Again, can't see the oocyte, don't need to, you know it's there somewhere. This little sticky outy bit that's about to burst is called the stigma. So the oocyte of this particular follicle and the granulosa cells of the corona radiata are going to come, oops, sorry, I accidentally tilted my screen, there we go, are going to come out of that stigma specifically. So unlike spermatogenesis, where the sperm are neatly deposited into the tubes of the epididymis, and then the epididymis delivers those tubes to the vas deferens and then on out, with ovulation, the egg literally just jailbreaks out of the side of the ovary, and then hopefully makes it into the oviduct. I'll talk more about that in lecture as well. It's fascinating, but I wanted to show you what that looks like. So that's pretty cool. Okay, so let's go elsewhere in this slide. Do I see any corpora lutea that I really like? Not so much. So I'm going to go to another slide for those. Ah, okay. Corpus luteum. Perfect. Okay, so corpus luteum means yellow body. Let's see if they were choosing the same one that I am. Yep, they sure are. Okay, so corpus luteum. It doesn't look yellow when you stain it with H&E, but if you were to take a fresh ovary and slice it, it would appear yellow. So I'm going to outline it in yellow in honor of its name. So ovulation, you can even see where that happened. So this part is the part that just closed off from being scarred due to ovulation. And then here is the antrum. And I'm putting an asterisk here because not all corpora lutea have an antrum. So as the corpus luteum progresses, if no pregnancy occurs, the antrum goes away, and the corpus luteum shrinks to become a scar on the ovary called a corpus albicans. So these cells, right here, these are the granulosa lutein cells. And so I'm not going to make you differentiate between them or name them. I'm just showing you what they're made of. So these used to be the granulosa cells secreting estradiol, and now they're going to secrete progesterone. So it's a terrible R, sorry about it. Progesterone. So the deal with progesterone, the short version, is the following. After ovulation, the goal of progesterone is to delay menstruation so that if conception occurs, the embryo has enough time to get down the oviduct and find a place to implant on the uterus. So progesterone not only helps to prepare the uterine lining for implantation of the product of conception, but also maintain it. So if conception occurs and an embryo implants, the corpus luteum is going to actually stick around until the placenta forms, and the placenta will then take over for the corpus luteum. If no pregnancy occurs, the corpus luteum gradually gets replaced with scar tissue, forming a scar on the ovary called a corpus albicans. Actually, I should do these in a different color probably, so let's do blue. So this one has a lot of corpora albicans. So blue are corpora albicans, and yellow is a big old corpus luteum. So corpus luteum means white body, corpus albicans means yellow body, corpus albicans means white body. So yellow body, white body, named for their gross anatomical appearance on a sectioned ovary. All right, let's take a picture of that. And then I'm going to actually zoom in on a place on this slide where the corpus luteum is right next to the corpus albicans, so you can actually see the difference. Okay, so over here in corpus luteum territory, we have a bunch of, so this area is highly cellular, and these are endocrine cells. Produce progesterone. Over here in corpus albicans territory, we have basically scar tissues. This is mostly collagen, not very many cells. So dense collagenous scar tissue. This tissue is mostly collagen and fibrocytes. So, big difference in appearance, big difference in function. The corpus luteum is capable of producing endocrine secretions that can sustain a pregnancy, and the corpus albicans can do none of those because it's just a scar on the ovary. That's all it is. Scar tissue does not have any endocrine function. So let's take a quick snap of that, and then we'll go on to the oviduct. Oh, if you're curious about what the ovary looks like right after ovulation, go look at this slide. Corpus hemorrhagicum, that big reddish thing that looks bloody on the left. But I'm not going to test you on it, so I'm not going to waste time on it right now. It's cool to look at, though. I'm not going to lie. Okay, let's look for oviduct. I'm going to pick this one. Yeah. Okay, so the oviduct here is really this. And the rest of what you're seeing here is connective tissue and blood vessels. So I'm seeing some arteries and veins embedded in dense irregular connective tissue, and then the oviduct itself. So let's zoom in on the oviduct, but first I'll label it. All right, zooming on in. Let's do it. So there's a thin layer of smooth muscle on the edge of the oviduct that has a name, and that name is, actually I'm going to use black. So this is the smooth muscle, and the name of it is the muscularis. And then instead of having a nice round lumen with no convolutions, in this case the mucosa is folded up a lot, as you can see. So just to give you an idea of what I mean when I say mucosa, I'm going to label it over here. So the mucosa equals epithelium, which in this case is ciliated columnar, but not for all mucosa. So mucosa is not specific to the oviduct. It's a naming scheme for layered tubes. So this will keep coming back in the urinary tract and in the digestive tract, etc., so forth. So the mucosa equals layers, the epithelium, and the lamina propria, which is usually loose connective tissue, and that is true in this case. So mucosal folds, which is on your list, are therefore these guys right here. So mucosal folds are those. Mucosal folds. And so are all the rest of the ones to the left of that. I just picked two to outline for the sake of brevity. So don't assume that those are the only mucosal folds you can see. They're just the two that I happened to pick, that's all. Okie dokie though. So there we have the parts of the oviduct, the mucosal folds, and the muscularis. And then really quickly before we stop, let's go ahead and zoom in on the mucosal folds themselves so I can show you the difference between lamina propria and epithelium. You can kind of see the cilia on here, but not that well. Well, I guess that's pretty good. So we have a columnar epithelium here with cilia. So this is one part of the mucosa. So we have a ciliated columnar epithelium, and then we have the lamina propria underneath. Which, as you can see, is loose connective tissue with the occasional blood vessel. There's one there. Here's an arteriole. So this is where the blood supply for that epithelium comes from. And then here is a closer up view of a mucosal fold. Alrighty, so that's that stuff. Thank you for your attention for these last couple of videos. And I am going to get busy posting these on the internet so that you can watch them. I'm also planning on recording a podcast today, so the second part of the endocrine system podcast will be out pretty soon, so look forward to that. Okay, thanks for your attention, and I'll see you in the next video, which will be gross anatomy of the reproductive system and the endocrine glands. Bye-bye.